Thank you, everybody, for participating in this week's workshop. I'm George Tiley, an evolutionary biologist. I am interested in many applications and questions in molecular evolution, but I am just particularly interested in this problem of what was the extent of Madagascar's grasslands in the past, and I think population genomic methods can be useful here. Here's a very familiar site to a lot of people in this group. Ankafo Bay, a small forest patch in the highlands, threatened by frequent fires. But it's also home to these cute little mouse lemurs. This is Microcebus latsara, the only mouse lemur found in the highlands. And in addition to these highland locations, can also be found in some patches along the escarpment, as well as some more lowland forests. So what are some things we can do with genomic data once we have it? Well, we can look at evolutionary patterns. Here's a species tree or population tree. We can start to say, well, the north diverged from the south first, and then after that, there was this splitting event between these eastern forest locations in the south and the highlands. And we can look at patterns of genetic structure. So the PCA really reflect both the tree and the geography. Here's Ankafo Bay and Tiley. Here are those eastern forests in the south, all clustered together. And here's that northern range. And then once we start to look at PC2 and PC3, we see, well, here's Ankafo Bay, Tiley, Ambatovi, which can be kind of indicative, perhaps, of a gene flow between these eastern forests in the south and the highlands. But something I like about molecular evolution is that we can often test hypotheses with probabilistic models. So, for example, a given demographic model, a fairly simple one, meaning that you can have population size change, splitting, migration. We can have expected distributions of the site frequency spectrum. They'll follow these sort of exponential distributions, and that exact shape depends on those demographic parameters. And once we obtain these multiple site frequency spectra, those can be used as input to evaluate different demographic models or look at population size change through time. The story that comes out from these mouse lemurs, when we look at Ankafo Bay, population dynamics are really not that different from other sites in the east. And if anything, all of these populations had a major decline after the last glacial maximum and through the Holocene, and perhaps there was another kick down after humans had arrived. Here's 1,000 and 2,000 years. Something interesting, though, that comes up when looking at patterns of gene flow is that there was migration fairly recently, at least as recently as we can detect, between Ambatovi, Tiley, and Ankafo Bay. So now it becomes really interesting to try and understand, well, what was happening with the vegetation around this time too. And here we are at 300 generations ago. And my background is initially in plants, so my intuition is to ask, well, let's have the grasses tell their own story. Can we gain something about grassland extent using these population genomic methods? And so far, we are focusing on a couple of open habitat species, C4 grasses, Aristida, Ludetia, and we're also looking at a shade-restricted understory grass, Aplis manus. But once you start working on plants, a number of complications can arise. One is that you just might not have the same genomic resources you do in primates, for example. The life history is complicated. We're not sure what a generation is. And of course, there's also polyploidy that confounds a lot of analyses. So my efforts over the past few years have been on overcoming at least some of these barriers. I'm a proponent of target enrichment data as an effective way to generate even population level data for many individuals without a reference genome. And it's also nice because it's not super dependent on the starting material, because all the libraries do get standardized to the same concentration. And then the main predictor of how successful your sequencing experiment is just how many reads you get, how much sequencing you do. The other area I'm primarily focused on is polyploid genotyping and phasing of those polyploid variants. There's a number of applications for coalescent methods where having phase data can be nice. And for a diploid, phasing is a fairly trivial thing. For the most simple case, two heterozygous variants, you can have 2 arrangements. For the most simple case with the triploid, there's 8 arrangements. And then of course, as ploidy and the number of link sites goes up, this gets very complicated. So colleagues and I have developed some software that uses heuristic methods to phase variants in a quick but effective way for polyploids. And we've shown this can be useful for applications such as estimating phylogenetic networks. Something I'm working on right now is designing some features into our software PATE, where you can estimate ploidy directly from the target enrichment data itself and generate these types of site frequency spectra so we can do some demographic modeling. Where do we stand now with our grasses? Well, we have some good population level sampling of those target species. We've managed to generate the data, and it looks good, but we still haven't moved on to any analyses yet. So fingers crossed that the results look good and that they're actually useful. Some limitations of the methods and population genomics in general. We have to make some strong assumptions about mutation rates and generation times to calibrate our analyses to absolute time. There's always danger of just getting the model wrong. And then of course, there can be genotyping error with polyploids that just don't really understand yet. I think there's a positive way to move forward, and together though, we really need some input work where everybody's coming together to actually develop what these models are. I think a real danger is we don't want to go on fishing expeditions or use too much circular logic and just focus on the molecular side. We need input from paleobotanists, ecologists, anthropologists on what plausible models are, and then we can use molecular data as a way to externally test those against each other. We can make some progress on resolving the taxonomy, because right now when we're talking about species of grasses, we're really talking about complexes, and there's some work there to be done. And in general, we can focus on collecting population scale data from many sites and many species. And at that point, we can really start to look at, well, what are broad trends across diverse groups of taxa that would be indicative of climate or island-wide effects. And along those lines, it could be useful to set standards for plant tissue collection. So we can even start to think about, well, what are some experiments or analyses that we want to do in 10 or 20 years from now. And with that, I'll just say all science, it's a team effort. I'm grateful for all of the people and collaborators that have helped me along the way.